Bismillahirrahmanirrahim. Today's lecture is on special topics in modern genetics, which will cover on various aspects of modern genetics, including DNA forensics, genomics, personalized medicine, epigenetics, and also stem cells. So we begin with DNA forensics. DNA fingerprinting is useful to establish a link between biological evidence and also suspect in a criminal investigation. And the biological evidence include blood, semen, hair follicles, or skin cells. And this technique is also used to establish paternity, whereby you can have a look at this link to see how this technique is used. Forensic science is the application of science to criminal and also civil laws, mainly on criminal side, during criminal investigation, as governed by legal standards of admissible evidence and also criminal procedure. The scientists will collect, preserve, analyze scientific evidence during the course of investigation. While some scientists travel to the scene of the crime to collect the evidence themselves, others will occupy a lab room and they will perform analysis on objects brought to them by other individuals. There are many tools that are available in forensic science, namely restriction fragment length polymorphism or RFLP, PCR for biological parent identification, random amplified polymorphic DNA for entomological investigations of corpses, for example from maggots, intersimple sequence repeat analysis, which is used to study genetic identity, parentage, clone, and also strain identification, and also for taxonomy studies of closely related species, gene mapping studies, mitochondrial DNA analysis, which is popular for maternal lineage identification, Y chromosome marker analysis for gender discrimination in rape and also in rape cases, and also paternal lineage analysis, allorepeat analysis to identify the population in ancestral lineage. DNA fingerprinting involves many intensive steps in order to fully complete and develop a DNA fingerprint, for example, of a father, a suspect, or a person involved in any criminal investigation or immigration problem. Okay, the process starts with isolating the DNA from any part of the body, such as blood, semen, vaginal fluids, hair roots, teeth, bones, etc. And then PCR is the next step in the process. In many situations, there is only a small amount of DNA available for this technique, and because of this, in a test tube, DNA replication is a must to occur to make more DNA. Okay? So this is where PCR is used. And after the DNA is isolated, more copies of the DNA have been made. The DNA will be tested. And the scientists will treat the DNA with restriction enzymes, which are enzymes that cuts DNA near specific recognition nucleotide sequences known as restriction sites. And this will further produce different size fragments, which are known as RFLPs. These fragments can then be observed on a gel electrophoresis, which separates the DNA based on fragment sizes. And gel electrophoresis in this process is the next step, whereby an electrical current is applied to a gel mixture, which includes the samples of DNA. And the electrical current will cause the DNA strands to move through the gel, and it will separate the molecules of different sizes. And the fragments of separated DNA are sieved out of the gel using a nylon membrane, which is treated with chemicals that allow it to break the hydrogen bonds of DNA so that there are strands and then single strands. Yeah? So this single-stranded DNAs is then cross-linked against the nylon using heat or UV light. And the probe shows up on photographic film because the strands of DNA decay and give off light. In the end, it leaves dark spots on the film. Which are also known as DNA bands of a person. And then what makes up the fingerprint are the unique patterns of the bands. And the pattern of the bands are different because we are all different and unique, other than identical twins. And once the filter is exposed to X-ray, the radioactive DNA sequences are shown and can be seen with naked eye. And then this will create a banding pattern, or what we know as DNA fingerprints. And this last technique is called Southern blotting. These fingerprints can then be used to determine which sample belongs to which person. For example, which hair strand belongs to which person. Yeah? And the DNA fingerprints of children should be similar to their parents' fingerprints, although they might not be the same. And some bands will match one parent, the other bands can match the other parent. With the bands of both of those parents, they make the bands and the identity of the child. So these are among the applications of forensic science, which include sample analysis, lineage analysis for suspect identification, anthropological studies, and also population genetic studies.